Welcome back. We are going to continue looking at special senses by taking a look at hearing in equilibrium. So, these two senses are going to exist within the ear, which we divide into three parts. We've got an outer ear that goes from the auricle here down to the tympanic membrane. We have a middle ear that is made up of this cavity here, and then we have an inner ear that is embedded in the temporal bone. The external ear and the middle ear are involved with hearing. The inner ear is going to be involved with both hearing and equilibrium. Now, the receptors for these two senses are completely separate. They respond to different stimuli and they are activated independently of each other. So they don't affect each other. You can have somebody who is completely deaf, they have perfect balance. You can have somebody who can hear great, but they don't have any balance. So they're not affecting each other. When one isn't working, it doesn't mean that the other one isn't working. So we're going to start with the external ear here. So we're talking about this area here. So the auricle is this outer portion that you're used to looking at. This is where we have the elastic cartilage. And then that's going to lead into the external acoustic meatus that runs into the temporal bone here. Here we are going to find ceruminous glands that are going to mix with the secretions to form cerumen, which is earwax. Earwax is going to help waterproof the canal and make sure that your tympanic membrane doesn't become brittle. That leads to the tympanic membrane, which is more commonly known as your eardrum. This is a very thin piece of tissue that is going to vibrate when the energy that are in sound waves travel down into it. The job of the external ear is to be a funnel. It is to take these sound waves that are out here and funnel them down to the tympanic membrane. The middle ear exists within this temporal bone here. It's called the tympanic cavity, and it has two windows. It has an oval window, and it has a round window. The oval window is sitting underneath the stapes here. And the oval window is basically the entrance into the cochlea. The round window is the exit from the cochlea. Energy is going to be transmitted in via the oval window, travel through the cochlea, and then back out through the round window. This is where we're going to find our ossicles. So these are very, very small bones that are going to connect the tympanic membrane to the oval window. You've got the malleus, the incus, and the stapes. And you can see they are very tiny, but they are bones. They have all the characteristics of skeletal tissue that we talked about before. The job of the bones is to take the energy that is pushing into the tympanic membrane. It's going to vibrate. The tympanic membrane will vibrate the malleus will vibrate the incus will vibrate the stapes, and then the stapes is going to push on this oval window that's lying behind it. We also have two protective muscles that we see in the inner ear. They are not shown here, but we can talk about their function. One is called the tensor tympani, and it is attached to the malleus. And when it contracts, it's going to pull the malleus inward, so toward the internal structure. The other is called the stapedius. It's attached to the stapes, and when it contracts, it's going to pull the stapes outward. So if you picture that all of these are connected, okay, you've got the tympanic membrane, vibrating the malleus, vibrating the incus, vibrating the stapes, vibrating the oval window. That's how the energy and sound enters into the cochlea, where you're going to have your really super delicate hair cells that are going to create the action potentials that are going to send information into the temporal lobe of your brain along the vestibulocochlear nerve. So there's a pathway to get that energy in there. If there is too much energy, so this is a very loud sound, it's a high decibel sound, that means that there's too much energy. And that energy is going to come barreling down the canal here and smash into the tympanic membrane here. Now, if we just let that happen, then the malleus is going to vibrate significantly, and then the incus, and then the stapes, and then the stapes is going to transmit all of that energy into the cochlea, and that's bad. That's how we damage our hearing. That's how the hair cells get damaged. So in the presence of a very loud sound, these two muscles contract and it pulls the malleus away from the tympanic membrane and the stapes away from the oval window, and it's going to dampen the amount of energy that's going to be able to travel through. So we do have some protection in there. That being said, modern day life, our ears have not evolved to deal with modern day life. We consistently are exposed to decibels that are going to damage our hearing. Walking down the street in New York City is above the level that we should be exposed to. So that kind of gives you an idea. If you live by the airport, you were never meant to hear those types of sounds. Like basically everyday life is damaging our hearing at this point. So the pharyngotympanic tube is also going to be found in the middle ear. So here you can see in a baby, this is the tube that runs from the middle ear into the nasal cavity, and here it is in the adult, from the middle ear to the nasal cavity. We tend to see in infants a lot of middle ear infections, and this tube is the reason. You can see that in the infant, it is very horizontal. It is not going to be hard for the microorganisms in the nasal cavity to gain access to the middle ear, where you really don't have a good immune presence. As your skull changes proportions, that tube becomes more vertical, and you're able to get a more well-developed flap of tissue in the nasal cavity that kind of covers the opening a little bit. The tube exists to help equalize pressure in the middle ear. When you are taking off in an airplane and you find yourself swallowing a lot, when you swallow, that little flap in the nasal cavity opens up and it allows pressure to be released from your middle ear. So the microorganisms, unfortunately, can move in there when that happens. But it is a lot more likely that they're going to be able to make this journey that is fairly horizontal versus this one that is fairly vertical. So the good news is that if you do have an infant that has consistent middle ear infections, they oftentimes grow out of them. So that's the good news. It doesn't mean that adults don't get ear infections, just not at the rate that we see in children. All right, the inner ear is a labyrinth. So it's a basically canal system. So we have a bony labyrinth that is filled with perilymph. So these are fluid-filled chambers. And then inside of that, if you picture like a big straw and then take a little straw and put it inside of it, inside of the bony labyrinth is a membranous labyrinth that's filled with another fluid called endolymph. So we have two fluid-filled chambers here. One of them is obviously going to be more flexible than the other one. So the bony labyrinth is not flexible. It is running through the temporal bone. The membranous labyrinth, though, is basically surrounded by a membrane. So that one's going to be more flexible. Within the inner ear, we have three different locations, starting with the vestibule. This is where we're going to find what it's called the saccule and the utricle that are going to house equilibrium receptors called the maculae. The semicircular canals are three canals. You have one for each plane of space. They are going to contain these sort of expanded areas called ampullas that house equilibrium receptors called the crista ampullaris. Last, we have the cochlea. That is the snail-like structure that contains three fluid-filled chambers that house your hearing receptor called the spiral organ. So here you can kind of see where this is sitting in terms of the temporal bone here. This is what it looks like if we take everything out. So here's your cochlea, here's the vestibule right here, and then here are your 3 semicircular canals. Here you can kind of get the feel for the bony versus the membranous labyrinth. So the bony labyrinth is running through the bone and is filled with the perilymph, which is shown basically in green here. Okay. And then running inside of it is a membranous labyrinth that is filled with endolymph, that is being shown in blue. So this is a tube within a tube. Okay. Now also make note that here is the oval window and here is the round window. So when we were talking about the idea of the entrance and exit of the cochlea, sound energy is going to come through here, go into there, it's going to pass through all of these structures, and then it's going to turn around and come out the round window. So if we look at the cochlea a little closer and talk about hearing. So the cochlea is basically these 3 fluid-filled chambers. And then it's very long and it's basically just rolled up on itself. Okay, that's where you get the snail. Kind of like if you have a sleeping bag, it's nice and long, but when you roll it up, it gets all rolled up into one little compact structure. So the chambers are called the scala vestibuli, that is filled with perilymph, the cochlear duct that is filled with endolymph, and the scala tympani filled with perilymph. So these two, the scala vestibuli and tympani, are part of the bony labyrinth, while the cochlear duct is going to be part of the membranous labyrinth. The oval window sits at the start of the scala vestibuli. The scala tympani ends at the round window. So picture entrance, exit for energy. Now, here are the fibers that make up the cochlear portion of the vestibulocochlear nerve. Okay, so basically running along the entire length of the cochlea, you have fibers coming in. And they are going to be associated with the spiral organ. So that's what we're looking at right here. So here are the fibers. They come in and they're going to attach to hair cells. Okay. And the hair cells have these projections called stereocilia that stick up, and they're in contact with a gelatinous membrane called the tectorial membrane. You can see it sticks out right there. When energy comes through here, it is going to cause this membrane right here, okay, the basilar membrane, this one right here, to vibrate, and it's going to push these hair cells up into the tectorial membrane, and it's going to cause them to bend. So here they are in their normal state, and then when they are pushed up against that fairly solid tectorial membrane, they're going to bend. That's going to open ion channels that are going to stimulate the action potentials that are then going to travel along these fibers of the nerve, sending that action potential along the nerve and into your brain. So the basilar membrane here is going to vibrate up and down, and when it does, it's going to push these hair cells into this tectorial membrane. They're going to bend, and it's going to send an action potential. So let's talk about sound for a minute. So sound is a pressure disturbance, and it travels in an S curve, or what's called a sine wave. So it looks like this. Okay, so that's how sound is going to travel. So there's a couple properties that are important in hearing. The first would be pitch. We can pick up different frequencies of sound. The higher the frequency, the higher the pitch. So let's look at this. Here is a time of, 0.01 seconds. The red sound had 2 waves within this time period, while the blue sound only had 1. So there is a higher frequency of the waves in the red sound versus the blue sound. So high frequency has these multiple waves within a given time. Lower is going to be less. High frequency is high pitched. Okay, you want to think of like that crazy high pitched scream that only a child can make. Okay. The other property is going to be the amplitude or the intensity or the loudness of the sound. Here's what we're measuring in decibels, and this has to do with the amplitude or the height of the wave. The higher the wave, the louder the sound. That's a direct correlation to how much energy is in the sound. Okay, somebody whispering next to you is not that much energy. There's not that much energy in that sound. Somebody taking a bullhorn and screaming into your ear, that is much more energy in that sound. So that would have a much higher amplitude of the wave. So we're going to get into this whole physiology. It's a pathway that we're looking at here. So this kind of breaks it down into the mechanics. So I kind of like this picture. You've got sound waves in the outer ear that are going to travel down the canal and hit this tympanic membrane that is going to be very flexible. And when the sound hits it, it's going to move in. It's going to begin to vibrate. It's going to push into the middle ear, which is going to vibrate the malleus, the incus, and finally the stapes. When the stapes begins to vibrate on the oval window, it is going to push in on that fluid-filled chamber, the scala vestibuli. So just picture taking your hand and smacking the surface of water. You're going to create waves. So there's going to be physical waves in the perilymph of the scala vestibuli. Those waves are then going to affect the endolymph in the cochlear duct because there's only a membrane between them. So, a membrane isn't solid. It's a very sort of flimsy structure. So if there's waves above it, that energy is going to transfer very easily into the cochlear duct where it's going to vibrate the basilar membrane. The vibrating of the basilar membrane causes the hair cells to bend. They run into that tectorial membrane. Now, as that same energy comes through the basilar membrane, it's going to end up in the perilymph of the scala tympani, where it will travel in the opposite direction and eventually equalize at the round window. So there's your oval window, there's your round window. All right, so let's go through this step by step. We've got sound waves that cause vibrations on the tympanic membrane, which cause the ossicles to vibrate in sequence. That is going to allow the stapes to vibrate on the oval window, causing waves in the perilymph in the scala vestibuli. Those vibrations travel through the vestibular membrane into the endolymph in the cochlear duct, where they're going to cause the basilar membrane to vibrate. It's going to push the hair cells against the tectorial membrane, causing the action potential. So at this point, we've sent the action potential, but we need to give this energy a way to exit out of the inner ear, or it's just going to keep stimulating these cells. So it's going to transfer through that basilar membrane into the scala tympani. You're going to get the waves in the perilymph there that will eventually sort of crash, if you will, into the round window, which will extend way out. It'll push way out into the middle ear, allowing a place for the waves to essentially die out. Just like waves on a beach, they run out of energy. The round window expanding out into the middle cavity is going to allow those waves to die out so that we're not continually activating the same thing over and over and over again. So here we can kind of watch this happen. So here's our energy hitting the tympanic membrane, and that's going to allow it to move through the ossicles in sequence. So the malleus, the incus, and then the stapes. The stapes then is going to vibrate against the oval window, creating waves in the paralymph of the scala vestibuli. Now, they're going to travel down to a certain point depending on the frequency of the wave, and then they will cross. So here you can see the length of the cochlea. So all along here, there are certain frequencies that are going to be picked up at certain areas. So each frequency has its own space to cross over. Now, if something is so low pitched that we can't hear it, it's below our level, it's going to travel through the scala vestibuli all the way down, and then it's going to go around what's called the helicotrema. It basically makes a U-turn and comes all the way out the scala tympani into the round window and equalizes, having never crossed into the cochlear duct. The hair cells are in the cochlear duct, which means that is something that we can't hear. So we are incapable of hearing something that is that low pitched. But most that we can hear are going to cross at a certain point. They're going to activate the hair cells as they cross the cochlear duct, and then they're going to travel in the opposite direction. This wave is going to come out through the round window, which will bulge way out into the tympanic cavity and allow the energy to dissipate. If we think about where this crossing occurs, all along the cochlea, you have a very specific area. If something crosses very, very proximally, it's a high frequency sound. Low is toward the distal end, and if it's too low, it just goes right around the helicotrema. Middle frequencies cross somewhere in the middle. So high pitch, high frequency is proximal, low pitch, low frequency is going to be distal. Now, the other property of sound is how loud it is. This has to do with how vigorously the basilar membrane vibrates. And that has to do directly with how much energy was in the sound. High energy sounds are very loud. They're going to come through, and they're going to have so much energy that that basilar membrane is going to vibrate really, really significantly. And it's going to be interpreted, that vigorous vibration is going to be interpreted as loud. Something that's really soft is going to come in with less energy, and the vibration is going to be much less, and it's going to be perceived as soft. This is where we have to be careful about sounds that have too high of a decibel. If there is too much energy in the sound that comes through, the hair cells can get pushed up into the tectorial membrane with so much force that they break. And these are neurons, so they're not going to fix themselves. This is how we damage our hearing. And we generally damage it at certain frequencies. So you'll lose a certain frequency that you had damage at that particular frequency from a certain sound because the hair cells were damaged. One of the worst things that we do is listen to headphones. I mean, we all do it, but when you listen to headphones, you're taking the energy that's in that sound and directing all of it directly into your ear. So there's nowhere for the energy to escape. So basically, we're all going to be deaf. Way earlier, than some of our, more recent relatives, simply because our life is just too loud and our ear has not evolved to deal with that. Okay, the good thing about equilibrium is that it functions exactly like hearing. So we went really in depth in hearing. We're not going to go too in depth in equilibrium. So you've got what's called the vestibular apparatus, consisting of the semicircular canals and the vestibule. The vestibule is going to monitor what's called static equilibrium, which is going to consist of the position of your head in space and the concept of linear acceleration. The semicircular canals deal with dynamic equilibrium, which is rotational types of movements. All right, so the maculae are what are found in the vestibule. So these are going to deal with your position of your head. So in other words, is your head perfectly straight on your neck, or are you leaning to the right or the left with your head? So that is going to be monitored by this, as well as the concept of linear acceleration. So picture you're in an elevator, the doors close. You know when you are moving, even though your eyes are staring at the door and it's not changing, because you can pick up this linear acceleration. You're sitting in a car at a stoplight. You've got your eyes closed because hopefully you're the passenger. When the car begins to move, you know it's moving without your eyes showing you everything going by. That's linear acceleration. Now, obviously, if I'm going up and down in an elevator or front and back in a car, that's two different planes of space. So the utricle deals with horizontal movements and the saccule deals with vertical. So what we have here is going to be the same setup. You're going to have hair cells that are going to be in contact with a gelatinous membrane, and when there is pressure applied to the membrane, the hair cells are going to bend and they're going to send an action potential. So in this case, the membrane, the gelatinous membrane, is called the otolithic membrane, and it has these little otoliths or little ear stones in them to give it some weight. So here, head up, here you're looking down. What's happening is that membrane is pulling, and all of those hair cells are bending because that membrane is pulling it. And that's going to send your action potential along the vestibular component of the vestibulocochlear nerve. The crista ampullaris is going to deal with rotational accelerations, and you've got one for each plane of space. So if I sit you on a twirly chair and twirl you around, that's one plane of space. But if I make you do cartwheels, that's another plane of space. And if I make you go off the diving board and twist in multiple different directions, that's all three of them. Again, same concept. Hair cells in a gelatinous membrane here. So each of your canals has this expanded area where you find the crista ampullaris. Here's your hair cells. Okay. The gelatinous membrane this time is called the cupula. When you begin to move, twirl, do something rotational, the endolymph travels in the opposite direction of the rotation, and it's going to push the cupula over, and that is going to bend the hair cells and send the action potential. So your sense of equilibrium is a pretty significant component. We've got not just your inner ear, but also your eyes, your joints, your muscles that are all involved in this concept of proprioception. Sometimes when these things don't line up, people can get kind of sick. So if you have what's called motion sickness, it happens when the information from your eyes is not matching the information from your inner ear. So for example, some people, if they sit in a car and they read a book, they get sick. They get nauseous. So what's happening there is the inner ear is sending a signal to your brain saying that we're moving, we're going forward. There is linear acceleration happening. Okay, your utricle is telling that. But your eyes are focused on the book, and the book is not moving. So your eyes are telling your brain, we are stationary. And your ears are telling them, we are moving. So generally, if you look up and you watch the scenery go by, then your eyes begin to match your ears. And suddenly we are not quite as nauseous. So it's definitely a incredibly important sense, but also fairly complex with all the different components that are feeding into it.